Now let's look at focusing, which mostly involves the lens. The lens's main function is actually to focus light onto the retina on the posterior aspect of the eye here. Now the lens itself is biconvex, meaning that both sides are convex towards the outer surface. It is also transparent, allowing for transmission of light straight through the lens and onto the retina beyond on that posterior aspect. Now, the lens itself is made up of columnar epithelial cells that actually then produce proteins called crystallins. These crystallins essentially act like a translucent layer, much along the classification of glass. Now, the lens itself is surrounded by a highly elastic transparent capsule. So, this elasticity is what allows it to become rounder when the ciliary muscle actually relaxes or tightens, and then actually become flatter as the suspensory ligaments and the ciliary body relax. So they are actually held in place by the suspensory ligaments here, and adjacently held to the ciliary muscle or ciliary body on the opposite side. Now remember that that ciliary muscle contracts and relaxes to change the shape of the lens itself. So if we look at how we actually do changes in shape of the lens to correct for objects being near or far, let's look at how that is projected. First, let's look at looking at distant objects. So this diagram up here on the right. Now, for distant objects, our ciliary muscle is actually relaxed, meaning that the ciliary muscle surrounding it is actually as large as possible, furthest away from the lens. This actually pulls on the suspensory ligaments, pulling them further away towards the sclera on each side, and it actually flattens the curvature on the anterior and posterior aspect of the lens, essentially limiting the convergence or convergence of the light from one side of the lens to the other. So this means like a tree far away from us, it comes in, and the light is actually relatively straight coming in. We're going to get a little bit of convergence here at the cornea, but based on it being so far away, the light is actually going to have a relatively small amount of change here at the cornea. As it reaches the lens, because that is flatter again, we're also going to have a limited amount of convergence, which will create our focal point slightly in front of the retina, and it actually will project the image upside down as well as backwards onto the retina, which will then be corrected upon when we actually interpret that information in the brain. If we look at how it works in close objects, so think of reading a piece of paper a few inches from your face. Now in this case we have the ciliary muscle contract, meaning it actually moves towards the center of the lens, decreasing the tension on these suspensory ligaments. The tension on those suspensory ligaments allow passively the lens to actually become more convex in shape. So greater curvature on this posterior aspect, more convex out here, greater curvature, convex here. Now, this means that more convergence is going to take place here at the lens, and so we'll actually have the ability to converge and create that focal point much further up within the center part of the eye, and actually project it again upside down and also larger, so as if we perceive that it is actually closer to us. So if we look at where the lens is again, remember that we have the sclera here, the white outer portion of the eye. We have the cornea, the clear transparent anterior portion. Here's the iris with the pupil, okay, this the muscle here that changes the size of the pupil, sphincter pupillae and dilator pupillae, and light passes through the cornea, through the pupil, and into the lens, where again the lens is changing shape based on this muscle here, the ciliary muscle, changing the tension on these ligaments, making it either more round or less round to again project that light back here onto the retina on the posterior aspect of the eye. Now, light itself is actually part of the electromagnetic spectrum, and it is a relatively small amount of the electromagnetic spectrum, only from about 380 nanometers to 750 nanometers. Outside of that, we have things like UV light, X-rays, gamma rays, microwaves, and radio waves, all outside of our visible light spectrum. Now, some terminology for utilization as we have light, the visible light passing through the eye is essentially convergence. We are always trying to use these convex surfaces, like the convex surface of the cornea, the convex surface of the lens, to actually create focal bringing together of light towards a focal point. The focal point is actually where the light rays converge and cross. And again, with more distance objects, we actually have that focal point a little closer to the retina so that that object will actually project smaller, as well as, again, upside down and backwards, whereas close objects will actually have the focal point slightly more anterior towards the lens and will actually project a little bit larger, but again, upside down as well as backwards. Both those corrections will be taken into account when interpretation from the brain takes place. Now, focusing is all really about creating that convergence point to a certain point so that we actually can change how our eyes focus on either a near object, a distant object, or somewhere in between. And so constantly we are using our ciliary muscle to actually contract and relax to change the shape of that lens, focusing it as appropriate to be able to visualize that distance. So here's that electromagnetic spectrum, and again, with very, very short waves, the distance between these peaks is very, very short. In gamma rays, very, very small, short nanometers, 1/1000th of a nanometer. As we get into X-rays, it's 1 nanometer to 10 nanometers. The UV light, again, from about 10 to about 100. Okay, and then we have the 380 nanometers to 750 nanometers being the visible light. On the opposite end of the spectrum, as the waves get much larger, okay, the space between the waves is actually where we get into infrared, microwaves, radio waves. And again, each of these, although maybe not be perceived by our eyes, can be picked up by other things like radio antennas or an infrared sensor. So if we look at focusing again, if we look at the far point of vision, this is essentially anything beyond 20 feet or more. So this is one of the reasons when we do our eye tests, we do a 20-foot distance, and usually it is given as not a fraction, but a proportionality to what 20 feet. So if you are 2200, that means that you can see at 200 feet, or what most people can see at 200 feet, you can only see at 20 feet. Now, this is the point at which essentially the lens doesn't have to actually thicken to actually focus. So it is actually as less convex as possible. It's really as flat as possible. Now, this is known as emmetropia. It is a normal resting condition of the lens. This means the ciliary muscle is relaxed. The tension on the suspensory muscles is actually, or the suspensory ligaments is actually very, very high, pulling that lens into an elongated shape, flattening that lens. Now, the near point is anything inside of 20 feet. Essentially, the lens needs to change in shape, becoming more rounded, okay, convex on each side in response to the change in closeness. Now, we also will limit the amount of light coming into the eye in terms of we'll actually shrink the pupil, so decreasing in size, and we'll also decrease the distance between pupils by actually medially rotating the eyes. So accommodation is essentially the ability for the ciliary muscle to actually change its contraction or relaxation. In this case, it is actually going to contract under the influence of cranial nerve 3, which is the oculomotor nerve, and this actually will pull this entire area here towards the lens, okay, and actually decreasing the tension on the suspensory ligaments, making that actually more round, more spherical. So it is biconcave, concave on the posterior aspect, concave on the anterior aspect. Now, convergence and pupil constriction again also occur. Pupil constriction, essentially we shrink the size of the pupil the closer things get, we enlarge it slightly the further away things get. Convergence, we actually will have the eyes rotate medially to accommodate for closer objects because generally the closer objects will be almost right in front of our nose, and so almost like going cross-eyed, but to a lesser extent. Now, as we look at the retina, we have two different main layers. We actually have the sensory retina, which is this area down in here. And then we have the pigmented layer, which is this area up in here. Now, the sensory retina actually is made up of three different layers of neurons. This layer up in here is actually where the photoreceptor cells are. These are actually cones, these are rods based on the structure conal shape and rod shaped. The next layer down in here is the bipolar cell layer. These are cells with essentially a single dendrite and a single axon extending from the cell body. And then we have ganglionic cells. These are these layers down in here, and these are the actual neuron cells that will then become the axons actually going into the optic disc and into the optic nerve itself. So the next layer down is the pigmented retina, and that's actually this area here. Now, this pigmented retina is actually a single layer of cells filled with melanin, and melanin is actually the pigment that is present in the skin, but it is also present to varying degrees within these cells, and it'll actually help to enhance the photoreceptor functionality, essentially reducing light scattering, but also enhancing color perception by isolating different colors within the spectrum. So if we look here again, we've got choroid, okay, just deep to the sclera. This is the pigmented layer of the retina, and then we've got a neuronal cell layer, or neural cell layer. These guys here again, these are actually going to be our photoreceptors, the cones that are actually going to do color, the rods that are actually going to just do light and dark. So night vision versus day vision. The next layer are the bipolar cells. And the bipolar cells, again, we have a single dendrite and a single axon creating a bipolar, 1-2 poles. It then connects into the ganglionic layer, which the dendrites receive the signals from the bipolar layer and send information out along the posterior aspect of the retina and all the way to that optic disc and then on to the optic nerve heading back towards the brain. So rods are going to be bipolar photocells, again, meaning that essentially we have a single axon and really a single dendrite. This dendrite out in here will actually be a photosensitive layer. The rods themselves are really for black and white vision, which really means night vision. So they're very, very sensitive to light, but they also will become damaged under too much of light instances, and so they'll actually shut off during day vision. Now, these guys are found over most of the retina, but not in the fovea centralis, where we're actually going to have only cones. And the fovea centralis is going to be our area of the highest visual acuity, our best vision. And remember that that's in the very, very center of the macula lutea. Cones, on the other hand, are again bipolar, so we have a single axon and really a single dendrite. Again, photoreceptive dendrite out here responding to light, but now in this case it is sensitive to color. It also gives us a higher visual acuity, and this is actually going to be all about the ability to pick up subtle differences between different colors, but also we will have less convergence, meaning that a single cone will often have a single bipolar cell, which will also then have a single ganglion cell associated with it, whereas rods, we tend to have a lot of convergence, meaning 4 or 5 or 6 even of these rods will actually converge onto a single bipolar cell, and many bipolar cells might actually converge onto a single ganglionic cell. Essentially, as the intensity of the light in here hitting cones actually increases, we actually will have an increased ability to see color. So rods, inside rods, again, we mostly do black and white vision, so mostly light versus dark, so it's night vision. These guys are very, very sensitive to light, but they also can be damaged, so they actually have developed an adaptation to shut down during conditions of extreme light. Now, the pigment present within rods is rhodopsin. This protein is actually two different parts. So we've got this area here, the larger part, which is the opsin, and then in here in the center is actually retinal. Now, in the dark state, meaning that there is no light hitting the rod, this is actually in a relaxed state, essentially what we call 11 cis retinal. Now, when we actually strike light into this cell, this retinal will actually change orientation. So we actually go from a cis orientation to a trans orientation. In this case, it looks like as if it's a horizontal or oblique orientation to a more fully horizontal orientation under stimulated light. Now, this retinal is actually then going to dissociate from opsin, from the greater opsin here. And so with sustained light, this retinal actually will detach and be completely separate from the opsin present just adjacent to it. Now, this retinal is actually derived from vitamin A, and so beta carotene, vitamin A are essentially what it is going to be broken down into when there is light present, and this makes the rod actually insensitive to further light stimulation. Now, because we have this change where we actually go from rhodopsin, the whole molecule here, to where we actually have an opsin plus a retinal, it means that it actually will take a little bit of time for this to recur and actually go back to having light-dark sensitivity, because we have to put the whole molecule back together again into rhodopsin each time we go from light to dark. So if you can think of the last time you went from a very bright environment to a very, very dark environment, it took a little bit of time for your eyes to adapt, and that's actually because we had to reform rhodopsin. Now, rods are a relatively unusual sensory cell. They actually depolarize when they depolarize when they are not stimulated by light. So when we actually have rods in a dark condition, the rods are actually depolarizing. So darkness stimulates the rods to depolarize. They actually will then release an inhibitory neurotransmitter here, and which they will inhibit depolarization of the bipolar cell. So this means that this one is depolarizing, the next layer is not. So in dark, depolarized, hyperpolarized, actually relaxed. Contrary, when we actually do have light actually hitting them, we have light hitting this cell here, and it actually becomes hyperpolarized, meaning that it actually will not release neurotransmitter. This absence of an inhibitory neurotransmitter in the synapse here between the rod and the bipolar cell will actually now cause the bipolar cell to spontaneously depolarize. This spontaneous depolarization will now release neurotransmitter and actually stimulate the ganglion cell. So this means darkness, okay, we get depolarized here, no release of neurotransmitter in a hyperpolarized bipolar cell, so that we get no signal going to the ganglion cell below. In a light characteristic, we actually get light, and this will actually cause this cell to hyperpolarize. No neurotransmitter that is inhibitory released here will then spontaneously allow the bipolar cell to depolarize, and as well as the ganglion cell to depolarize. Now, rods themselves, again, we have rhodopsin broken down into opsin and retinal. So again, it makes it less sensitive to further stimulation of light, but it also takes time for it to actually reaccommodate. Pupils, on the other hand, can respond relatively rapidly. When we actually go into a very, very bright environment, we have constriction of the sphincter pupillae, and the pupil will actually narrow, limiting the amount of light in a very light environment. Or when it becomes very, very dark, we have the dilator pupillae here that actually constrict and pull the pupil open, allowing more and more light. So we have two different light adaptations essentially based on long-term light-dark stimulation versus short-term. Cones are, again, going to be a bipolar cell, but again, now these are responsible for color vision. They actually will, the more intense the light is, the more likely we are to be able to see differences in lights or in colors themselves. Now, cones are really, really high concentration within the macula, so the densest portion of the retina, but the highest and only we will have in the fovea centralis, we'll have only cones. So this is the area where we get our highest visual acuity, essentially the ability to see the most differentiation. Now, another difference between rods and cones is the pigment utilized. In cones, it is iodopsin. This is actually have three different pigments that are actually present within cones, and they'll actually respond to blue, green, and red light, and their overlap and essentially their light spectrum allows us to see many different gradations of color. In theory, millions are possible. So if we look at the inner layers of the retina again, remember that we had this pigmented layer here, we have the rods and cones, we have the bipolar cells, and we have the ganglion cells. Now this is looking at the convergence. If we look at rods, we have 5 rods up in here, okay? And these 3 actually converge onto a single bipolar cell where we have 1, 2, 3 synapses, all from these 3 rods onto a single bipolar cell, and this is known as convergence. This bipolar cell then synapses onto this ganglion cell here, which also is synapsed onto by another bipolar cell that is fed into by 2 other rods. So rods have lots of different convergence, meaning that 3 synapse onto 1, 2 synapse onto 1. These 2 also only synapse onto 1. So stimulation of any of these 5 rods will actually only fire a single ganglion cell. Cones, on the other hand, where we have a higher visual acuity, we have a single cone here, and each of those cones synapses onto a single bipolar cell. These bipolar cells, each individually, again, attached to a single ganglion cell, this one to this one, this one to this one, and this one to this one. So we have very little convergence. Again, a 1-to-1 ratio of essentially cones, ganglion or bipolar cells as well as ganglion cells versus a 5-to-1 ratio between rods